us, everybody. My name is Raj Prasad. I'm on the biotech team at William Blair. I've got my colleague Sandy Corwin joining me as well. And we've got four esteemed CEOs of gene therapy and gene editing companies who are hopefully going to help me kind of figure out what's going to happen in the space over the next 10 years. And I appreciate each of you for taking the time out of your day, and hopefully everyone's staying safe and healthy in their respective homes. To start, maybe just a level set, would love kind of everyone, each person to take a two minutes to kind of give a high-level overview of your company and kind of key catalysts that investors can expect over the next, say, 12 to 24 months. We'll start with Karsten from Selecta.
Thanks, Raj. Yeah, my name is Karsten Brunn. I'm the CEO at Selecta. Selecta is an immune tolerance platform company. Our technology is called ImmTOR. It's a biodegradable nanoparticle that encapsulates rapamycin. It's infused, given together with an antigen, induces T regulatory cells specific to an antigen. So antigen-specific immune tolerance, what we do. We have a couple of applications. One is to improve the efficacy of biologics. We're about to enter phase 3 in a rare inflammatory disease in Q3 this year. We also have a readout in a head-to-head trial versus the currently approved product in that market. We also, and that's why I'm here on this panel, work on enabling redosing of gene therapy. The plan is to combine ImmTOR with an AB capsid. And so in this case, the antigen would be the capsid. And we have some pretty compelling preclinical data that demonstrates that we can retreat, and we plan to enter the clinic in MMA, methylmalonic acidemia, in the first half of next year.
Jim?
Hi, everyone. This is Jim Burns. I'm the CEO of Locana Bio, and we are a RNA-targeted gene therapy company. So what we're doing is combining what we think are two very highly validated approaches to genetic medicine, gene therapy, as well as targeting RNA. We're doing it differently than I think what others have done in the past. So we're actually delivering RNA binding proteins that can be engineered in different ways to affect dysfunctional RNA in different ways. And I think that's the strength of our platform, is that it's not just destruction or just splicing modification, but there's actually 5 different ways that we can affect RNA. We can destroy normal RNA as well as repeat expansion RNA. We can destroy and replace. We can enhance translation and haploinsufficiency. We can do RNA editing as well as splicing modification. We're mainly focused in neuromuscular disease as well as neurodegeneration and retinal disease. But as the platform develops, there's a number of other disease areas that we can ultimately move into. Over the next year or so, what we see is that probably by early next year, we'll be in IND-enabling studies in multiple programs, most likely in the neuromuscular and the neurodegeneration space. And hopefully, if all goes well, then within 2 years, we'd be in the clinic.
Okay. John?
Great. So thanks for having me, Raj. Looking forward to this. So I'm CEO of Beam Therapeutics. So Beam is in the gene editing space, working particularly on some next generation systems for gene editing, particularly something called base editing. And so what we do with base editing is we use CRISPR in a different way to target genes and make single letter changes in those genes. Is that me? Sorry, everybody. Stop that. Don't know how. All right, there we go. So base editing makes single base changes in genes, so down to a single letter of DNA. We can change those letters without cutting them. And so that turns out to be a real advantage to do different kinds of things, such as correcting point mutations being the largest class of genetic mutation that drives disease. We can also use them to silence genes or reactivate genes. And so we're taking the precision of that approach and trying to apply it very broadly. So we're going after a variety of different therapeutic areas, all of the major delivery modalities we're pursuing in parallel. So we have a pretty broad pipeline with a lot of different kinds of programs moving forward quickly, but certainly also building on the great progress that's been made in the CRISPR field. by Sam and CRISPR and others who's come before us. This year, we're publishing a lot of in vivo data out of the programs. We'll be hopefully naming our first development candidate out of the pipeline, and then looking next year to begin with IND filing.
And Sam?
Thank you, Roger, for having us on this panel, and it's great to see all the other panelists. Jim, hopefully you're enjoying the San Diego shores more than the East Coast, we're pleased to be here, and I hope everyone's staying safe. As Roger mentioned, I'm the CEO of CRISPR Therapeutics. We're close to 5 years after formation of the company now and setting up operations here in Boston, and we're very excited and pleased to be able to move this platform, which we all knew had tremendous potential, but to move it into the clinic and see some early promising data. Essentially, as we think about our company, we have 3 parts to the company. One is our hemoglobinopathies franchise, where we have early data from CTX001 in a couple of thalassemia patients and 1 sickle cell patient, where we saw promising data that could potentially lead to a cure. We have our immuno-oncology franchise, where we have 3 different CAR Ts in the clinic targeted towards CD19, BCMA, and CD70, both in heme malignancies as well as solid tumors, and we're enrolling patients across all 3 of those CAR T programs. And then we have the rest of the pipeline, which we don't oftentimes talk about, but we have a very promising regenerative medicine program targeted towards type 1 diabetes, where we edit pluripotent stem cells and differentiate them into islet cells. We're doing this with our partner ViaCyte, and a number of other programs where we use viral and non-viral approaches to edit genes in vivo in the target organ of interest. So we look forward to moving those programs along to the clinic as well. And we're now close to 350 people at the company and growing rapidly as we try to advance all these programs.
Great, thanks everyone for those overviews. It was very helpful. I kind of want to switch gears now and talk about in vivo delivery, which seems to be a huge hurdle for both gene therapy and gene editing. So could each of the gene editing companies explain their preferred approach and what they see as successful clinical editing rates as opposed to the numbers that we're seeing ex vivo? And are there additional safety parameters taken into consideration when comparing in vivo and ex vivo? And, so I guess not to exclude you, Carson, but we'll start with Jim.
Yeah, well, you know, actually, Sammy, in some ways, we're not really a gene editing company, but we have some of the same challenges, right? So what we're doing is actually using gene therapy to deliver payload that would then manipulate RNA. But we have very similar challenges with respect to delivery. But what we're doing is taking what's already been successful, for example, AAV9 for delivery to the muscle, and there are certainly serotypes that you can use for delivery to the CNS and into the spinal cord. So our first approach is to take what is already there and make sure that we can adapt it for what our current purposes are to show proof of concept. At the same time, we're also looking at what could be next in terms of more efficient delivery, better tropism, dealing with what everybody that delivers with AAV deals with in terms of immunogenicity as well. But one of the things I wanted to mention also is that what we have discovered with the systems that we've developed is that they're highly potent. So what we're finding, for example, in our animal models for DM1 is that we're getting, depending on method of delivery, anywhere from 30 to 90% of the muscle fibers actually are transduced. And what this allows us to do is actually lower the dose for what we ultimately would deliver, which has tremendous implications in a very positive way in terms of potential safety, because dose, especially for systemic delivery, is may be related to safety in the patients ultimately. So, I think, what we have now can be useful, but there's a lot of room for improvement, obviously, as if we're going to expand the application of gene therapy and gene editing and what we're doing with RNA targeting.
Great, thanks. And John?
Sure. Yeah, so I think clearly ex vivo, like Sam, we are also in the hematology field and doing cell therapy by editing T cells. You can get, 90, 95% editing there because you can put your machinery directly into the cells. That's really exciting. In vivo, no question, delivery remains the biggest challenge. But again, it's 2020, like a lot of progress has been made. So there we're doing both lipid nanoparticles to get to the liver, but you've seen great progress from Moderna, from Alnylam, from Intellia on that front. So we're not reinventing the wheel there. Same thing with viral vectors like AAV, as Jim said. We know a lot now from the gene therapy field of where those can go, and we just put the gene editing equipment into it. One of the nice things about gene editing, of course, is that it's a one-time therapy. So in some ways, our bar is a little lower than gene therapy because we don't have to create therapeutic levels of a transgene for life. We just need to get enough editor in to edit enough of the cells to create a threshold for cure. Ultimately, obviously, the level you need to get to is going to be different disease to disease. But anyway, bottom line, I think the field has moved along a lot, and we think that although each of these technologies could improve and we need more, there's plenty to do now with what's already clinically validated in the delivery space.
And Sam?
Yeah, I think I agree with what Jim and John said. I think they covered most of the points. I'll just say I think it took longer, but it's going to get figured out. I think we're going to see a number of programs in the clinic with in vivo delivery and gene editing or RNA editing that are going to come to bear relatively soon. I think the only point I'll add is it does take a little longer because you have to move species to species. In ex vivo settings, you typically can go from a mouse experiment to humans with the right PK/PD characterization. Whereas in vivo, you typically have to at least do a couple more species, and those tox studies and those studies take a long time to do. So, in general, the length of time and investment is a little greater for an in vivo therapy, but once you crack the code, it becomes relatively scalable.
Great. Just a quick follow-up to this discussion, and then I want to bring Carson in. What do you think about, maybe John or Sam, what do you think about the length of time you'd need for a delivery vehicle? I mean, there's discussion of having it be a little more transient of a delivery device because it's a gene editor. Maybe just a little bit about the dynamics there. And then after that, maybe Carson, you can discuss on the potential for redosing AAV and how you think of it in competition with gene editing.
I can lead off. I mean, I think so. Certainly, duration is a consideration in delivery. So with AAV, you can have long-term duration, although that's not true in all tissues, actually, right? And so that's something that we think about. But certainly for an editor, you don't need long-term duration. So we would want to think about off switches or things like that if that was going to be the case, depending on the tissue. LNPs, obviously transient delivery, delivering an RNA and guide RNA payload. We like that. That's a perfect fit for editing. That also can enable redosing, which is attractive. And so we're certainly excited about that application.
Yeah, but I think, in our experience, the edits happen very quickly. So you don't need long-term expression at all. I think there are certain cases where, if you're delivering sort of donor templates and through an AAV, you may need to wait a little bit longer so that the episomes are formed and the donor templates are expressed. But in general, I think you're talking about a very transient expression, and that's the advantage of gene editing. It's one and done, and I'll bet you, I know right now, if you think about the market for DMD or hemophilia A, people assume that gene therapy is it, right? Gene therapy is the answer. And I think unless Carsten comes up with a solution, these things are going to be only 3 or 4 years. I mean, they're not going to, and especially as you dose younger patients, they're not a lifetime therapy. And I'm particularly excited in hemophilia and DMD, where, or DM1 for that matter. I think if you go with the gene editing approach, as long as you get to a certain level of editing, I think they can be a lifetime cure, even with the very transient expression of the delivery vehicle up front. And that I think could be a superior product in the marketplace in the long run.
Yeah, that's a nice segue, actually, Sam. As you rightly said, one of the limitations of, especially of AAV-mediated gene therapy, is that once you're exposed to the vector, you produce very high levels of neutralizing antibodies. So you can't give a second dose. At the same time, AAV vectors don't self-replicate, and especially in organs like the liver, and especially in pediatric diseases, the liver actually grows 20-fold from birth to adulthood. The vector gets diluted out. But even in adults, in Hene, if you look at the Hene 4-year data that's been out there now, you see a decrease in expression of the transgene, and those patients have to go back to effective replacement therapy at one point, right? So what we try to do, obviously, and we have pretty compelling data preclinically, where we combine our technology ImmTOR with an AAV vector, and we're able to do actually 3 things. One, we're able to prevent the formation of neutralizing antibodies, which enables a redosing, at least in animals. And then maybe even more importantly, we've shown that the transgene get expressed in the second dose in the presence of ImmTOR. So we're actually able to express the transgene in the second dose. And the third finding, which is interesting, and we'll have to see whether it translates into humans, but we also see a first dose benefit. If you combine ImmTOR with an AAV gene therapy, you get up to 4-fold higher expression of the transgene, actually, which could potentially mean that you have to give lower doses, which helps with safety and also with cost of goods as well, obviously, because that's the bottleneck right now in AAV gene therapy is the manufacturing. So obviously, we have demonstrated technology in other indications where the antigen is a pretty immunogenic non-human enzyme, and in this case, now gene therapy would be the AAV capsid as the antigen. So that's the approach that we're taking. We have a number of partnerships. We'll talk about that later, I think, Raj. But maybe on the question of gene editing versus gene therapy. So we haven't done any work in gene editing, but obviously the Cas9 protein is bacterial origin, and it's also immunogenic as well, actually. So there might even be potential applications of ImmTOR in that field. But I think just gene therapy is further along with a couple of products on the marketplace. But gene editing obviously is the more elegant approach, potentially more durable as a one-time treatment, and also more targeted. And especially I think what you're working on, base editing, or the next I think evolution is prime editing. It's getting really targeted where you can do single mutations. You can exchange basically a single base pair. So I think that's obviously a lot more targeted. But I think from just looking from a commercial perspective, I think there's currently, 900 INDs out there in gene therapy. So obviously, there's going to be a whole wave of AAV-mediated gene therapies where retreatment is an issue. So I think from our perspective, at least over the 5-year horizon, this is, definitely there's a more urgent need. 5 years plus, depending on the data from gene editing, that landscape might change, obviously. But I think from our perspective, next to the pre-existing antibodies, which I think are also an issue in gene editing, the retreatment probably is the most urgent need you have currently with AAV gene therapy.
So switching gears a little bit, in January, the FDA released its final cellular and gene therapy guidance document. So I was hoping each of you could discuss briefly how those guidance documents apply to your company's technology and pipeline indications, and how the guidance will influence the preclinical and clinical development, as well as primary endpoints, as well as long-term follow-up, which seems to be a concern for the FDA. And we can start with Sam.
Thank you for that question. I think I'd like to commend the FDA on coming up with the guidance, which is quite helpful for all the companies. I think the European regulatory authorities had put something out in the 2016 timeframe, a little bit ahead of the FDA, but this guidance from the FDA is very helpful for companies, especially with the number of INDs that were mentioned earlier. For me, the biggest takeaway from all the guidance is two things. One is they use the words variability and quality repeatedly in the guidance, I think. And a lot of that concern around variability comes from all the viral vectors used for manufacturing and the subsequent products, even if it's CAR Ts that are manufactured. And then quality obviously is of paramount importance because, as the cliche goes, the process is the product, and it's, oftentimes you're getting different products for different patients. But if you don't have the robust quality systems. And so I think it's actually very beneficial for gene editing and RNA editing, if I use the term loosely, in the long run, because what you're going to see is a much tighter distribution of the drug product and the manufacturing process with gene editing, as opposed to a stochastic distribution that you see with viral therapies. That is of great concern to the regulators, in addition to obviously all the other parameters that look at in a deep way. But those would be the big takeaways for me, is that robust manufacturing with the highest quality level is important, and ensuring low variability in the drug product is paramount in getting fast regulatory approval.
Carsten, do you have anything to add?
So I think obviously we very much look at redosing and we're looking at the rare disease guidance. The FDA actually specifically mentions the need for repeat administration and the need for assessment of immunogenicity. So I think it's an acknowledgement of the FDA that companies need to look at ways to be able to retreat, and I think that's quite a step forward. If the topic wasn't on topic for a long time, and now it's officially in an FDA guidance document, I think kind of underlines the importance actually of retreatment.
Jim, do you have anything?
Well, probably everybody in the field was probably using the draft guidance documents that go back a number of years. So it's actually nice that it's been codified, and we officially have the final versions of what we are going to practice. So I think in some ways it's business as usual, in the sense that we're going to continue to use what we assumed was going to be there. We were happy to see that there wasn't anything new from what was already drafted there, but also, that for us, it was important to see what the long-term follow-up would look like for use of AAV. That certainly is consistent. As long as they're not modifying the genome itself, then it looks like it's about 5 years. It's going to be 5 years for long-term follow-up. So I think it's very helpful. Like Sam said, I'm glad that it's there. Europe was a little bit ahead of us here in the U.S., but I think it's going to be very helpful for us as a roadmap going forward.
And John, what are your thoughts?
Yeah, I won't add much to what was said. I think the headline for me is that this is a rapidly maturing field. I don't, people might still think of this as like the Wild West. It's not. As 900 open INDs in gene therapy is astonishing, and there are increasing numbers of INDs in gene editing now. So, our experience has been that regulators know what they're doing. The assays have really come along, so we're able to bring that high science, that qualification and rigor that Sam mentioned to the regulators, and I think that's what they're looking for. And we know the playbook. We know that manufacturing is critical, that quality is critical, delivery matters. So I think, I think it's all part of the, gradual growth of this whole field.
Great. And, we'll come back to platform and preclinical, clinical development, but I'm an analyst that has to give the people what they want. So John, let's switch over to business development for a second here. Maybe talk about your business development strategy, your wholly owned pipeline right now. Very good worded question by an investor that I'll just repeat. Could you speak about the idea of potentially partnering with the CRISPR company, and what benefits would that offer to you?
Yeah, well-worded question. We wouldn't generally comment on any market activities or anything like that. That's not what we're in the business of doing. I would say more broadly, to answer your question about our strategy, again, Beam has benefited a bit by being a sort of a new entrant to a field that's already made a lot of progress. And so that's enabled us to rapidly build out what we're doing. One of the decisions we made early is maybe in contrast to what some of the other CRISPR companies have done initially, was we haven't done a lot of partnering. And we made the decision to keep our assets wholly owned. For us, that means that we can control them, we can move them into the clinic, we can generate data, and then we can make a decision at a later time point about what we have, what the competitive landscape looks like, where we will choose to grow versus where we think we need help. So obviously we've got 2 programs in sickle cell, we've got some CAR T programs, we're working on alpha-1 antitrypsin deficiency. So these are huge opportunities and very complex spaces. So we really like the idea of moving those under our own steam forward, and then at some point we will certainly make that decision about what kind of partnerships make sense. As for relationships with the CRISPR companies, so we have, of course, our collaborators with and licensees of Editas. That's a great relationship. I've known Sam for a while now. We're close with Intellia as well. I think it's a closely knit community, I think we're constantly looking at opportunities, but mostly we are focused on developing base editing, developing internally the capabilities that we need to turn this into what we believe is a potentially important new class of medicines.
Yeah, thanks for answering that question, John. Sam, on BD also, I think you've done a fantastic job with being able to monetize some of the non-core assets that you have, or the ones that you haven't focused on first, I should say. Can you maybe just talk about your BD strategy as it relates to some of the deals you've done more recently, as well as even the deal that was initially done with Vertex as well for CTX001?
Yeah, I'm happy to talk about that, and also point out that the world has changed a lot between 2016 and where we are today. I think in the 2016 timeframe, gene editing was still very nascent and early, and the equity markets were looking for pharma validation in a sense. I think if we were in a different permissive environment, I think in general it's probably better not to partner your assets too much, because you certainly monetize it, but at the same time you're slowing down things, slowing down your programs, and it's not every big pharma has the capabilities to add to a emerging field like gene editing. So I think given the environment we were in, the strategy that we had was to create that validation, monetize some of the non-core assets. But I would say that the one exception is the Vertex partnership that we have in CTX001. If you think about that program, that certainly has benefited from having Vertex as a partner in a 50-50 fashion, because we've moved that program along rapidly, and as we scale up and we think about global clinical trials, it's difficult for a smaller company to have a footprint across the world, to make those capital investments. And I think having Vertex as a partner on the lead program has certainly helped us quite a bit. And that was the logic of doing the deal on DMD as well, which is DMD is going to require a lot of scale up. These are higher doses of AAV that you're talking about, and I think having a manufacturing scale at that level requires significant capital investment. So from a capital efficiency standpoint and an ROIC standpoint, I think it made sense for us to do most of the buy-side deals that we did. But right now our focus is more the sell-side deals that we did, but now the focus is on buy-side to essentially acquire technologies in support of our programs to enable us to be even more successful and to move our programs along faster. And buy-side was one example, and we're looking at other potential opportunities. We recently licensed RegenMed technology from the University Hospital Network in Toronto, Canada, and we're looking at other deals as well.
Great. And Carson, maybe you can just describe some of the BD deals you've done recently. Obviously, a couple intriguing gene therapy deals with Sarepta and AskBio. We'd love to kind of talk about your BD strategy and what you see as your go-forward kind of in-house pipeline.
Yeah, I mean, obviously we're in a bit different situation than Sam. We're not a gene therapy company per se, we're an immune tolerance company. We do have an in-house proprietary program in OTC deficiency, but we have two partnerships. So one with AskBio, many of you will know, a private gene therapy company based out of North Carolina. And really, what AskBio brings basically the manufacturing piece to the collaboration, right? I mean, that's a hard thing to get access to. And we have a strategic collaboration with them 50-50 where we develop a number of indications and commercialize together. First one being MMA. And all the data we have generated so far is the liver-based diseases. And obviously, the deal with Sarepta is a research agreement where they basically want to replicate the experiments we have done in liver-based diseases to be able to retreat in neuromuscular disorders. Did a deal for DMD and a number of the limb-girdle indications as well. So for us, they're enabling, obviously, financially transformational. But I agree with the comment that obviously it's preferable to have your own assets, as that we have one in-house. And once we demonstrate a retreatment, we can definitely also look at in-licensing programs as well. Because if you look at the gene therapy field, there's a lot of companies pursuing very small indications, and there can only be one or two winners. And if we're able to differentiate by redosing, there's also for us to actually in-license assets as well. So definitely the goal is to have our own proprietary programs.
Jim, I'm going to pull you back in here. I know Locon is private, so you can't discuss BD as much, but with the data generated to date with DNA editing, how do you see RNA manipulation fitting into the treatment equation?
Yeah, that's an interesting question. So I might actually turn it around just a little bit in the following sense, that we know that RNA is a valid target, that there are ASOs as well as siRNA products that have been able to correct dysfunction through either deletions or splicing modifications. So I think it's pretty clear that RNA is probably a rich target area that's untapped. And I feel that there's going to be a lot of different ways which one could attack dysfunctional RNA. I think gene editing for sure is going to have a place as a genetic medicine. There's no two ways about it. And down the road, I think I alluded to this before, I think there's going to be a toolbox of genetic medicine so we can think about what is the right way to attack a disease. And hopefully at some point we'll move beyond what can I do with my technology, right? But for now, that's what we have to think about. So the fact that there are dysfunctional RNA approaches, there is a fact that there are dysfunctional RNA approaches that lend themselves to some very interesting systems for being manipulated. So let me give you an example. And Sam can probably appreciate this from my Casivia days with something like autosomal dominant retinitis pigmentosa. So it's a toxic gain of function approach. There's 150 mutations in the rhodopsin gene for which you could potentially have an approach from a genetic engineering standpoint, gene editing. or through manipulation of RNA, you can have an approach where you just knock out the rhodopsin message RNA and then you replace it with a normal piece of RNA or DNA. So I think there's a lot of examples like that where you can say that it's complementary. So I wouldn't say that it's necessarily competitive. I think at the end of the day, you find what's the best way to approach the disease and what's the best, so what's the best approach for that disease in terms of different genetic medicines.
Do you think there's a set of indications that would be more depth to being targeted with RNA manipulation rather than gene editing?
Well, I tell you, one of my favorites is in the microsatellites repeat expansion diseases, like CUG repeats for DM1, CAG repeats for, there's 20 of those, Huntington's disease, ataxin-1, ataxin-2, there's several of them. C9orf72 for familial ALS. And one of the beauties about attacking it at the RNA stage is that you can attack the repeat itself and you're not attacking the actual product of the gene. So you leave the wild type in place, but you only hit the wild type. So you're only destroying the wild type transcribed DNA. So I think that's an advantage, potentially an advantage. I mean, data will tell us whether that's the case or not. But I think that's a really interesting approach.
Thanks, Jim. This one's for Sam. Sam, obviously, you got some clinical data coming up for CTX110 later this year. Can you maybe level set for us on what to expect with that data set? And in particular, maybe just describe some of the nuances between editing HSCs versus T cells. And then maybe a broader question, maybe include John in this one, about different nucleases and your thoughts on that as well.
Yeah, happy to start. I think on CTX110, this is our allogeneic CAR T targeted towards CD19. I think you've seen data sets from our competitors, or peer companies, I should say, more so Allogene and Precision Biosciences around a similar target with allogeneic CAR Ts. We're doing a phase 1/2 study, which is evaluating safety and also the doing a dose escalation. So I think what we're hoping to see in this trial, one, is establish that allogeneic CAR Ts can be safe, safely administered. But then we want to see, what are the doses where you start seeing activity, and then how does that compare to autologous CAR Ts? The bar you're looking at essentially is the CR rate from autologous CAR Ts. Durability is, is going to take more time to establish with allogeneic CAR Ts, but at least on the early stage, are you getting responses that are comparable to autologous CAR Ts? And if they are comparable, I think, it's going to be very difficult for autologous CAR Ts to make a dent going forward, because it's so much easier to administer allogeneic CAR Ts to patients. So that's sort of what we're looking for from the data set. We're encouraged by the data set from Allogene and what they showed as responses. There's a lot more to learn, but so far, I think we generally feel good about our hypothesis around the allogeneic CAR Ts. Editing HSCs versus T cells, I think there's an art and a science to all this. I think the T cells in general, if you think about editing rates 3 years ago, they were single-digit percentages, and through a number of changes over time, we've now gotten to a point where we can get 50-60% editing rates with over 10 edits at scale that we can do in these CAR Ts without translocations or compromising viability. Obviously, as base editing and other technologies emerge, it kind of adds to the versatility of a CRISPR-based platform. And so we're going to have more and more sophisticated constructs going after these cancers, and that's a good thing for patients as we advance the field with better constructs, which you can do, by the way, with autologous. Is very difficult to improve on early data. With allogeneic, there's significant improvement potential.
Great. And anything else to add on different nucleases or the toolbox of nucleases and how that may play a role in kind of next gen or different indications? Anybody want to take that?
Yeah, I'm happy to start. And John, I'm sure you have some thoughts there. But I think we are, there's many different ways of improving the nucleases, right? I think a lot of the comparison originally was based on sensitivity and specificity. And what's clear now is with CRISPR-based editing tools, you can actually get very high efficiency in a very specific fashion. I think where we're now going to is speed and versatility. Can you get very fast cycle drug development with the CRISPR-based platform, and then can you do multiple edits? Because we're finding more and more as hundreds of labs work on these cells in terms of edits that can be made to improve the cell viability, robustness, persistence, et cetera. And I think all the new platforms will come into play, whether it's better nucleases, smaller nucleases, but base editing and potentially other editing forms as well.
Yeah, I mean, so Beam is not primarily a nuclease company. We do own a nuclease family called Cas12b, which we are working on. But I think maybe to broaden the question, as Sam said, I do think these systems are so powerful because they're engineerable. So you can tune them, you can change them. And then particularly in the CRISPR systems, just by changing that guide RNA, you're retargeting your whole system to a new section of the genome. You have an entirely new medicine. So the incremental investment required to go from target A to target B and get a new program is really quite minimal. So although the platform investment up front is a lot, the ability to create a sustainable pipeline for the long term will be pretty high. For us, we worked initially on escalating fetal hemoglobin in sickle cell disease. We got to 95% editing there. We moved from there to directly targeting the sickle mutation. There's a single point mutation that causes that disease for all patients. We now have editors that can 80% edit that disease-causing sickle mutation. That editor turns out to share a lot of DNA technology with the editor we're using for alpha-1 antitrypsin deficiency. Totally different mutation, totally different part of your genome, and yet a lot of the same principles applied, and now we have editors there that can edit that disease-causing mutation directly. So I think as these tools, diversify and become more sophisticated, the applications expand as well.
Right, and then maybe just a quick follow-up, John. Obviously, Dave Liu put out that review where he had a huge flow chart in one of the figures. I mean, how are you thinking about how you utilize the base editing platform kind of juxtaposed against that figure, and also if you guys could just comment on prime editing, as it's kind of like the new kid at school. So
yeah, sure. So as David Liu had a review on base editing. It's a field that has sort of exploded since it was first begun in the spring of 2016. A lot of that, complexity now of how do you create base editors that have different targeting ability, different specificity, different efficiencies. That's all what Beam is designed to do. So our know-how is really deep there, of course, and we've really pioneered that field. We've published advanced versions of our own of the C base editors and the A base editors in Nature earlier this year. We're only at the beginning. I mean, there's going to be a lot more coming there. I think there are other technologies as well that are interesting. Prime editing is another. So I think this general theme of trying to avoid cutting the DNA if you can, and then doing different applications will be, I think, a big part of the growth of the field in the future. With base editing, if you just want to change one base, at this point we can do that with pretty high efficiency, great specificity. So I think if you can get away with a single base change, base editing is probably good, and that's all you need. But there's other things you'd like to do, and so prime editing, for instance, may enable short-range rewriting of DNA, and so you can do, multi-base sort of signature changes, and that again came out of David Liu's lab, and we're using that technology along with a new entity that's being started to exploit that as well.
And I know we're just about out of time here, but since the title of this panel is Gene Therapy in 2030, I feel like it'd be remiss if I didn't ask where you each saw both your companies and the field in 10 years, with the obvious caveats of your safe harbor statement. So if you could just quickly discuss that, starting with Carsten.
10 years a lifetime for public companies. So, but I think so from a Selecta perspective, obviously, and maybe we're talking 5 years, we obviously hope to be an established player in gene therapy, enabling redosing, enabling dose finding, and which is a key challenge right now, with a number of products on the marketplace, from a vision perspective. I think longer term, I think 5, 10 years, I really think, and it was mentioned earlier that, genetic medicine will be mainstay of therapy. I think we're going to move away from treating symptoms to, treating underlying diseases, I think, which is fantastic. And, I think gene therapy will still be around. I think there'll be gene editing. And I think also if you look further out, I mean, synthetic genomics is kind of the next big thing. So I think it's pretty exciting. And I think it's good for all of us as potential patients, actually, right? And because you're really making a profound, actually, impact in patients' life with those. And I think we sometimes obviously forget this when we talk about the science and evaluations, but I always find it, especially in genetic diseases that have a profound impact actually on patients. So I think, I'm quite optimistic for the next 10 years for this field.
Jim, what do you think?
You know what, I think that in 10 years, I'm hopeful that we're going to have better delivery systems, because if we have better delivery systems, then I think a lot of things change. And I think we will have genetic medicines that are going to provide cures for patients. I think that we'll have genetic medicines that can be one-time treatments. Maybe they won't be cures, but they're going to change people's lives in dramatic ways. And I think that the key to that is obviously understanding the biology around what we're trying to do, but it's going to be delivery.
John or Sam, care to close us out?
I'll let Sam close us out. So I'd say I disagree. I mean, I think there's a little, we differentiate among each other, but I actually think the big trend here is what percentage of the therapeutics pie is going to be one-time genetic therapies in 10 years. And I think it's going to be way more than it is today. I think there's lots of drivers for that. It's good for patients. It's good for families. It'll be good for hospitals, and it'll be good for payers, actually, because there's real value there. So, I think all of these technologies are going to play a big role in the future. And so for Beam, we want base editors to be one of those classes of medicines, and we want to be the drivers of that. And we think we can create a real sustainable wave of innovation out of this technology.
Yeah, and I'll agree with all what my panelists said. I think in a dozen years, I bet a third of the pharma market will be cell and gene therapies or non-small molecules and non-antibodies. And that's a significant growth over that time frame. And I think our aspiration, and it's very hard to say where we're going to be. I mean, there's a lot that needs to go right, but our aspiration is to be the next Genentech. Genentech took 25 years between 1980 when recombinant proteins were discovered to becoming a $100 billion company, and I think we hope to do it in a faster time frame. But the goal is to build a cutting edge and organization that's going to continue to innovate and create breakthroughs for many different diseases.
Well, great. I share all of your sentiments on the future of this space. I'm excited to cover it and also work with all of your companies. And I appreciate each of you taking the time to chat about this exciting area, and hope everyone's staying safe and healthy, and look forward to chatting soon.
Thank you.
Raj.